Hey, 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 this video is designed to help you with your microbiology exam 2 at Gadsden State Community College. And so let's jump right in. The title and the topic of this first PowerPoint is all about bacteria. So I know on exam 1 we talked about some fungi and protozoans and some viruses, but exam 2 really gears in, really the rest of the course, but specifically exam 2 really is talking about bacteria. So remember that a prokaryote is a cell without a nucleus or without membrane-bound organelles. So we're talking about bacteria, E. coli, strep, staph, things like that. I pulled this picture out of an old high school textbook I used to teach out of. I just thought it was really cool to see the difference in size. And so you can see the virus right here. And then the bacteria is here, and then this would be like just an average human cell, maybe like a cheek cell or skin cell or something. And so you can see that bacteria are much smaller than human cells, but viruses are way smaller than anything else. Here's the difference in a prokaryote and a eukaryote. Remember, this whole test is predominantly about prokaryotes. That's cells without a nucleus, without all these mitochondria and we're talking about prokaryotes. But you can see that prokaryotes are much less complex than eukaryotes. And remember from exam 1 when we talked about classification, all bacteria and only bacteria are prokaryotes. So animals, plants, fungi, and protists, that means algae and protozoans, they're all eukaryotes. Okay, so most of this you're going to already know. These are just features of the prokaryotic cells. They lack membrane-bound nucleus, so they don't have a nucleus or any of the other organelles. We haven't mentioned this yet, but they don't have these special proteins called histones that are associated. When you see a chromosome on commercials or whatever, it's shaped like an X. Because of the histone proteins, animals can make these X-shaped chromosomes. Well, bacteria don't have those chromosome proteins. They don't have histones. So they do not have histones. So they do not make X-shaped chromosomes. And they have a cell wall made of peptidoglycans. So we mentioned that on the first test when we did gram staining. And they reproduce by binary fission. So they make identical copies. And we'll talk in class about some other ways that they can reproduce besides binary fission, but for now just know that they reproduce asexually. They make identical clones by binary fission. Here's a picture of eukaryotic chromosomes making that X, and you can see the green histones there. Bacteria do not have those. Prokaryotes do not have histones. Here's a picture of binary fission. It's simply just the cell dividing. It's very simple. It happens rapid. This is similar to what you learned about in a previous science course, maybe a high school science course, about mitosis. So binary fission is just bacteria's more simplistic form of mitosis. Okay, so just different ways that we will discuss how to differentiate in this PowerPoint. Oops. The shape, so if it's round coccus or rod-shaped bacilli. The arrangement, how do they grow? Do they grow in chains or clusters? Do they have external structures like flagella? What is their chemical composition? Do they have a special chemical like the glycolic acid that's found in the organism that causes tuberculosis? Biochemical activities and energy requirements and nutritional needs kind of fall under the same thing. What do they eat? What do they need to survive? Things like that. So that's just ways we'll discuss prokaryotes. This is a picture. You can see these bacteria are round, and they are in a line, and so we would call this streptococcus. The arrangement is strep. Strep is in chains. Coccus makes it round. Okay, so coccus we know is round. We saw that on the first test with gram staining. Whoops, there we go. Okay, coccus is round. If it's in clusters like grapes, we call it staphylococci. So what I would do if I were you, I would draw this on my picture out beside staphylococci. Well, I guess it's on the next page, but I would draw that out. Staphylococci, and then streptococci, I would make a chain. I know it's not very scientific, but I can remember that strep throat is in your throat, and you wear a necklace chain on your throat. And there's way more streptococcus than the organism that causes strep throat. But to remember that it's a chain and strep. Anyway, two of them is diplococci. Look at this picture here. And then you can see this one is the tetrad. The top right is tetrad. And then Sarcini is 8. So it kind of looks like a Rubik's Cube to me. So you do want to know those. You see strep, staph. And then the next set of them are the bacilli. So you look at the words, and I'll talk about the picture. There's single bacilli. 2 is diplo. DI is 2, right? Diplobacilli. Streptobacilli is strep is in change, remember? And then coccobacillus is, they kind of look chubby, don't they? They're like rods, but they're a little bit chubby. They're rounded. Okay, and then we talked about some spiral organisms, and these are really interesting. Okay, so there's Vibrio. It's a common shaped curved rod, and it causes, if ingested, it's fecal contaminant. And all you have to know is what's on the slide, but it makes it a little bit more interesting if you know what it is. But cholera is it's an infection that you actually get from fecal contaminated water. It was huge before the United States got clean drinking water. Thank you for clean drinking water. But it causes bad diarrhea. So it's fecal contaminated water. Spirillum that's a shape of organisms. That is a corkscrew. Think about it like a wine opener. So a corkscrew shape. And then a spirochete is more flexible, kind of like an Arby's curly fry. You can see the picture here. You can see the difference in the spirillum and the spirochete. Now, there are 2 examples of spirochetes that you've got to know. Borrelia burgdorferi is Lyme disease. Tryponium pallidum is syphilis. So the organism name is Borrelia burgdorferi, and the disease that it causes is, or the infection that it causes, is Lyme disease. And we know how we get that, right? We get that from ticks. So if you're ever bitten by a tick, the rash will come up. They call it a bullseye. A rash will come up, and you want to keep an eye on that and make sure you don't get a bullseye rash. And if you do, you want to go to the doctor and get a round of antibiotics, because if not, Lyme disease can be very serious if not treated. Tryponium pallidum is a sexually transmitted disease which causes syphilis. Weirdest thing about syphilis, it doesn't have very many side effects or symptoms that are in the genitalia region. Very mild symptoms. You might not even notice that you had any symptoms, but it travels through to the nervous system, to the brain, and it destroys by corkscrewing into parts of the brain, destroying brain tissue. And so someone wouldn't know they had syphilis until they realized that they had a brain problem. Okay, so most bacteria only have one shape, and we call that monomorphic. But some bacteria can go back and forth. They can switch shapes, like go coccus bacillus, they can switch back and forth. 2 examples are Corneum bacterium and Rhizopium. So those are examples of pleomorphic. So on the test, make sure you know the examples. So 2 examples of spirochetes are listed there, Borrelia burgdorferi and Tryponium pallidum. 2 examples of pleomorphic organisms are Corneum bacterium and Rhizopium. So make sure that you know those examples for sure. And there's a picture of a spirochete under an electron microscope. There's some more pictures. Okay, some bacteria can make this sticky substance on the outside thick, sticky substance is produced within the bacteria and then excreted to the surface, and it helps them survive. It's simply that. It helps them survive. If it's a slime layer, which is loose, or a more tight layer, which is a capsule, and it says that encapsulation increases virulence. That's how sick you are. So you have a higher fever, you vomit more. The symptoms are worse if there is a GLYCOCALYX layer. The capsule protects the bacteria from phagocytosis of our immune system. So you remember macrophages, how they identify foreign particles, maybe from anatomy. If not, it's okay, but they identify bacteria and they engulf them and destroy them. Well, the capsule, kind of like the envelope with the viruses, it helps it to be camouflaged from our immune system. Okay, so this is all about that capsule. The capsule doesn't stain. You have to use a negative stain. So the background would be pink, and then the bacteria would appear colorless. So we'll look at some pictures of that, or maybe slides in class. But it's like the whole slide is pink except some white, and that's where the organism actually is. Okay, so you want to know these functions. You want to be able to pick out which one is not a function on the test. We said it increases virulence. It helps the bacteria stick to surfaces. You want to know the examples. All the examples are going to be on the test. An example of an organism that has a slime layer capsule is Streptococcus mutans. It sticks. It allows the bacteria to stick to the enamel of our teeth and cause cavities. Number one bacteria associated with tooth decay, cavities. So the slime layer allows it to stick to the enamel. It's used for nutrition. So the bacteria, if it gets hungry enough, it can eat its own capsule. Silly, I know. And then it also helps the bacteria not dehydrate. So it's almost like a chapstick around it or something. Okay, this is just showing you the difference in. This is showing you the parts of the flagella, I think. Let me see what's next. Yeah. So you can so the flagella is a whip-like structure, and you can look at the picture and see that whip-like structure that helps the organism propel forward, so move. So bacteria that move do so by flagella. So the ones that we're talking about don't have cilia, they don't have pseudopods like the protozoans we discussed. So bacteria that move do so by flagella. There are some really, really strange ones with some cilia, but don't think about that. Bacteria that move do so with flagella. Okay, so parts of a flagella. These are just basically some vocabulary words. If a flagella, and I would draw these in, has one flagella from one end. If it has flagella from both ends, like that, and that can actually be more than one. You want to know these pictures. You may see these pictures on the test. Okay, wait, I messed up. Okay, sorry. Flagella. So Amphitrachus is flagella from both ends. I did mess up. It can be more than one. So one or more. So both pictures would be correct. Okay, if it has a whole bunch from one end, and then peri, P-E-R-I, means around. So flagella all around. And so you want to be able to identify those pictures. You may see those again. Okay, when you look at the flagella, the long extended part, like the hair-like structure that we really think of when we think of flagella, that's called the filament. The hook is the wider portion right next to the cell wall. And then the basal body are these rings that anchor inside. So basal body, ring pairs which attach to the flagella. So, and the flagella, it spins like the propeller on a boat. It doesn't go back and forth like a fishtail. It does it spins so that it can move the bacteria forward. So look at this picture. You can see the filament. That's the long extended portion. You can see the hook right next to the cell wall. And then you can see those ring pairs, which we call the basal body. Okay, only organisms that are spirochetes have axial filaments. Only organisms that are spirochetes have axial filaments. Okay, so think about when you took Anatomy 201, you talked about actin and myosin. Those were filaments that allowed muscles to contract, and they slid back and forth. Do you remember that? Okay, well, it's okay if you don't. But these axial filaments, they slide back and forth similar. It reminds me of muscle contractions. But it allows a spirochete to do that corkscrew movement. So we talked about two spirochetes, Borrelia burgdorferi and Triponium pallidum. So they have axial filaments. Let me show you a picture. See how it goes, it's up under the sheath, and it runs down the spirochete organism, allowing it to make that corkscrew movement. And so, let's see, spirochetes only, a bundle of fibers that extend from one end underneath the sheath to the other. Fibers turn from within the cell and give it that spirochete movement. Okay, these are on the same slide, but they're both short hair-like structures. Okay, so FIMBRIAE are used for attachment. The example here is Neisseria gonorrhea is the organism. You want to know the examples. It allows the organism to stick to the mucosal layer of the vaginal wall. So we already talked about Streptococcus mutans can stick to the enamel of the teeth because of a capsule. Well, Neisseria gonorrhea can stick to the vaginal wall because of these little hair-like structures called fimbriaes. Okay, PILIs are also hair-like structures, but they're used for reproduction. This PILI stretches out, and bacteria can exchange DNA. Unfortunately, they normally exchange the DNA that's associated with antibiotic resistance. So some bacteria, for example, MRSA, is resistant to penicillin. If you don't take all of your antibiotic, the PILI can be attached to other organisms, and they can share that segment of DNA. So other organisms can also become resistant to antibiotics. And that's why they tell us to take all of our antibiotics. Okay, endospores. Oh, these are so interesting. Endospores is where I'm going to show you the picture first. The organism can take all of its important parts and wad it up in the middle, and then that other outside part, literally, it just falls off. And it's almost like hibernating. The organism can go into this endospore form, and you can see it there. That last little orange piece is the endospore. And it can survive a long, long, long time, or high heat or dehydration, as long as it's in this endospore form. So your textbook tells us that endospores can survive 19 hours of boiling water. So they can survive pretty much anything. You can't tell me that a Lysol wipe will kill an endospore if 19 hours of boiling water won't kill an endospore. Come on, right? And so highly resistant. It can survive almost anything except an autoclave, open fire, things like that. We'll talk later about some things that it can survive. Can survive almost anything. All endospores are gram-positive. You want to know that. Gram-positive, except there's one that is negative, gram-negative, and we call it Coxiella. The two best-known endospore formers are Bacillus and Clostridium, and they're gram-positive. So right there, I've given you 3 examples of endospores. Bacillus, Clostridium, and the gram-negative Coxiella. Bacillus and Clostridium are gram-positive, and Coxiella. Bacillus, think of the organism that causes anthrax. You can see it on like the TV shows. Bacillus anthracis, that is a bacillus. We'll use some bacillus in lab also. And then Clostridium is, just for example purposes, it's the organism that causes tetanus. So we know about vaccines against tetanus and all that. But on your bonus assignment, it tells you to list 3 ways that we discuss Clostridium, and this is the first one. Clostridium is an endospore former. When they become the spore, we call that sporulate, and then when they come out of their spore, we call that germinate, and it can happen almost instantly. So when you cut the hot water on, and it before, like, you know, right as it starts to boil, they can endospore and survive. Okay, talking about the cell wall, we've mentioned peptidoglycan several times now. Peptidoglycan several times now. So the cell wall is made of peptidoglycan. It prevents pressure changes. It's hard, it's rigid, it maintains its shape. It helps some bacteria cause disease, and then it also helps us differentiate. Does it have a thick cell wall? Does it have a thin cell wall? Does it have mycolic acid in the cell wall? Okay, gram-positive versus gram-negative. You know this from exam 1, but we're going to hit some new topics here also. Gram-positive organisms have a thick wall of peptidoglycan. Okay, this is new. They have a trace amount. That means just a small amount of teichoic acid. Resistant to physical, think about like brushing. Okay, think about we're gonna do a lab where we use a UV light. So write in UV light is a physical control. Gram-positive are resistant to physical controls. That means resistant means they are not easily killed by the UV light, which is used in food buffets, used in surgery rooms, ORs, operating rooms, or like think about the wiping of your hands. And then they're sensitive to chemicals. So that means like when you use Germ-X, you know, you use the Germ-X and then you rub your hands together. Okay, the Germ-X, the actual chemical, is going to kill the gram-positive. Sensitive to chemical control. The rubbing is not going to kill the gram-positive, but that's going to kill the gram-negative. So think about the ways that we clean. We most often clean with a chemical and a physical. When you mop your floors, you physically rub your floors, but then you're also using a chemical, like a Lysol or something like that, a vinegar maybe. When you brush your teeth, you are brushing, that's the physical, also using a chemical, which is the toothpaste. Okay, gram-negative has a thin cell wall. It also has this fatty layer on the outside of it, which offers extra protection and makes them sometimes more difficult to kill. Phospholipid, lipoprotein, lipopolysaccharide layer. So think of it as like an oily, greasy layer on the outside of that thin gram-negative. Sensitive to physical, resistant to chemical. So instead of trying to memorize this resistant chemical stuff, try to understand it. The gram-positive thick cell wall is hard to brush and kill, but a chemical could eat through that peptidoglycan. A lot of antibiotics target the peptidoglycan cell wall, like penicillin. Penicillin works by breaking down the peptidoglycan cell wall, so it is effective on gram-positive. Penicillin is not effective. It is ineffective on gram-negative. So pause. I recommend pausing this slide for just a second and really trying to understand instead of trying to memorize, trying to understand it. The gram-negative, it's got that greasy layer on the outside, so it is resistant to chemicals. It always makes me think of that a Dawn commercial that says it breaks through grease. You know, so I guess it takes a chemical like that. Not all chemicals mix with grease and break it down. There's a picture you can see on the right-hand side. That green layer, that's phospholipid, lipoprotein, lipopolysaccharide layer on the outside of the gram-negative. On the left-hand side, you can see a thick peptidoglycan wall. And then a little bit, those, I don't know, was that orange circles? That's going to be the trace amount of teichoic acid.